Hello students, welcome to 60 out of 60 in KCET Chemistry 2025 series in just 200 days. Today we are going to do Biomolecules part 2, where we'll be covering the proteins and enzymes. Okay, so in our previous class we have covered carbohydrates, so today we'll be doing proteins and enzymes, and then we'll be practicing a lot of MCQs. Okay, so please sit with a notebook and a pen and note down all the important informations. Now, one more request, please join our WhatsApp channel. The link is there in the description section. You can go there and join our WhatsApp channel, join our WhatsApp channel, and this will help you to get access to all the PDF notes, the mock test, question papers, solutions, everything you can easily access. Okay, so please do not forget. Now, without further delay, let's get started. Okay. So, amines, as we are doing today part 2, and we'll be covering proteins and enzymes today, right? And in the next class we'll be doing vitamins, nucleic acid and hormones. Okay, so weightage, as I told you, it will be like 2 to 3 questions you can easily expect from this chapter, and it is very easy to score this 2 to 3 marks because this chapter is like completely you have to mug up some data. So just you need to study, revise and learn this data and then directly apply and solve the questions, right? Next, in proteins we are going to discuss today what? Amino acids, classification, structure and then denaturation of proteins. Okay, so let's start. Now let us discuss what is amino acid. See, like carbohydrates are made up of one single unit called monosaccharide. Similarly, proteins are made up of these particles or these units called as amino acid. Okay, many amino acid combines to give you protein. Okay, now what is the structure of this amino acid? So amino acid consists of 2 important components, that is, you see amino is there, that means amine group will be there, and acid is there, so carboxylic group is there. Okay, and then carbon, hydrogen and one R group is there. Now based on this R group, based on this different R groups, we have total of 20 amino acids. Okay, so here are the name of different amino acids. You can see every amino acid has this common. There is NH2, there is NH2, there is COH group. Alright, you can see that every amino acid has this structure, and now these R groups are different for different amino acid. Based on the R group, the different 20 amino acids are there. So example like glycine, alanine, serine, then you have threonine, cysteine. Okay, so these are different amino acids. Now coming to, so there are total 20 amino acids, no need to remember the structure for them. Okay, so now something called as Zwitter Ion. Okay, so about this amino acid, it is told that they are behaving as salt. Okay, now when is a salt formed? A salt is formed when an acid and a base react, right? So in the amino group, in an amino acid, you saw there is both basic group. Which is basic group? The NH2 group, because it has lone pair of electrons, and an acidic group, that is COH group on the same molecule, right? Both of these are present on the same molecule. So there is an intramolecular, you can say, proton exchange. So what happens? This H is released and this is accepted by this NH2 group to give you this one. So this becomes COO minus and this becomes NH3 plus, right? So now what is this? This compound is neutral because the overall charge is 0, right? But it contains both negative and positive, and this is amphoteric in nature. Okay, this is N. Now why will it be amphoteric? Because it has both basic and acidic group, right? So this particular compound is called as Zwitter Ion. Okay, so Zwitter Ion is a form of amino acid where it exists in the dipolar form, one negative and one positive charge. Overall it is neutral, but yes, it is amphoteric in nature. Okay, now coming to classification. So see, classification of amino acids. So on the basis of nature, we have 3 types of amino acids, and one we have on the basis of synthesis. Now on the basis of nature, we have acidic, basic and neutral. Now what is acidic? You see there are 2 groups, right? COH group and NH2 group, and there is a side chain. So if in the side chain there is more COH group, then we will be calling it as an acidic amino acid because more carboxylic groups are there in the amino acid. Now in the basic means if there are more NH2 group, then we'll call it basic, right? Now let us see one example. Just a minute, we'll check first. Yeah, you can check here now. Okay, you see aspartic acid. This is NH2COH, which is common to all the amino acids. Now you see there is one extra acidic group here. So this makes this amino acid acidic in nature. Okay. Similarly, you see in glutamic acid there is an extra COH group, right? Now if you see for basic group, you can see glutamine. Glutamine, you see there is an extra NH2 group in the side chain, right? And then you can see aspargine. Aspargine has also an extra NH2 group. So these groups make these amino acids basic in nature. Okay. So that is there. That is your acidic nature, basic nature and neutral. Then coming to synthesis. Synthesis is very important because on the basis of synthesis, we have essential amino acid and non-essential amino acid. Now what is essential amino acid? That which cannot be produced in human body. Okay. If it cannot be produced, then how are we going to get this amino acid? From the diet, okay, from the plants, from the food we need to get these amino acids. These are called as essential amino acids. Now non-essential can be produced in the body, okay, can be produced in the body. Clear? Now we'll see the example. Out of 20 amino acids, there are 10 amino acids which are essential. Okay, so I'll tell you the list of now, you can remember. So we have valine, leucine, isoleucine, arginine, lysine, threonine, methionine, phenylalanine, tryptophan and histidine. These are the 10 essential amino acids. And one more very important thing, please remember these 10 amino acids. Okay, essential amino acids example, please remember. Okay, next coming to peptide linkage. Now I told you what, see, proteins, we discussed about proteins are made up of amino acids. I told you in the first line when I discussed about amino acids. So what is this? When these many amino acids are connected, they form proteins, right? Now these amino acids, this is amino acid 1, this is amino acid 2. So the bond between these two amino acids is nothing but peptide linkage. You can call it peptide bond. Okay, peptide linkage or peptide bond is that bond which connects or which is formed between two amino acids. Okay, now how this bond is formed, let us see. See, you have NH2 group and you have COOH group. This is amino acid 1. This is amino acid 2. Here also we have NH2 group and here also we have COOH group. Now here what happens, there is dehydration. Okay, so one OH from here and one H from here is removed. So what is left here? CO is left here and NH is left here. So that is what is present, and this is nothing but your peptide linkage. Okay. So this amino acid is your glycine and this is alanine. So we call it glycyl alanine, GLY, and written ALA. Okay. Next. Okay. Now when these amino acids, okay, this small, like suppose there will be many, there can be many number of amino acids combining. Okay. If the number of amino acids is greater than 10, then that particular chain will be called as polypeptides. Polypeptides are those where the amino acids are more than 10. Okay. Now a polypeptide with more than 100 amino acid residues having molecular mass higher than 10,000 is called as protein. More than 10, polypeptide. More than 100 and mass is also around 10,000 or more than 10,000, then we will call it as a protein. Okay. So you can see proteins are those chain of amino acids where the number of amino acids is more than 100 and the mass is around more than 10,000 U. Okay. Now, what is a dipeptide? Okay. Now see, if two amino acids are there, then we call it a dipeptide. Tripeptide means there will be 3 amino acids. So in a tripeptide, how many peptide bonds will be there? there will be 2 peptide bonds in a tripeptide. Now if you see like for example tetrapeptide, okay, means tetra means there will be 4 amino acid, okay. How many bonds will be there? 1, 2 and 3, okay. So the number of amide linkages will be less than what? How many amino acids will be there, okay. This is very obvious. Okay, now we'll go to the next thing, that is structure of proteins, okay. So structure of proteins can be like, one thing we are going to study is molecular shape. What is the shape of the protein? Next we'll study about the structure of protein. So in molecular shape we have fibrous proteins and globular proteins, okay. Some of the proteins are fibrous, some of the proteins are globular, okay. So this classification is done on the basis of the molecular shape, okay. Now coming to structure, so we have primary structure, secondary structure, tertiary structure and quaternary structure of proteins, okay. So this is not a classification, okay, I am telling you this is not classification. About structure, when I am telling you, the protein structure can be discussed in the 4 levels. There will be primary structure, secondary structure, tertiary and then quaternary, okay. And if you talk about the shape of the proteins, it can be of 2 types, fibrous and globular, okay. Now let us discuss them in detail. First thing, that is the molecular shape, fibrous and globular proteins. We'll discuss the examples here. So in fibrous proteins, what happens? The polypeptide chains run parallel and are held together by hydrogen and disulfide bonds. So there are like polypeptide chains, they run parallelly and these are held together by what? Hydrogen bonds and disulfide bonds. Now here what happens, polypeptide chains coil around to give a spherical shape. So there is coiling of polypeptide chains here, okay. This is insoluble, this is soluble. Some examples you can see, keratin and myosin. Keratin is found in your hair, wool and silk. Myosin is present in muscles. These are your fibrous proteins. And globular proteins example, insulin and albumins, okay. So this difference is asked many a times in the exam, so in your PU board, so you need to remember here, okay. Next we can just see the structure of proteins. The first one is primary structure. Like I told you, the level will be in 4 levels, primary, secondary, tertiary and quaternary. So what does the primary structure of protein tells you? See, primary structure is the sequence of amino acid, okay. So I told you there are 20 amino acid, okay. So these amino acids can be arranged in many different ways. So the sequence of amino, like for example, you made a amino acid like alanine, then you mixed glycine, then you made valine, then you did histidine, right. So this sequence of amino acids in a polypeptide chain is called as primary structure, okay. So any protein will have a primary structure where there will be the sequence of amino acids. If you change this sequence, the protein will also change, okay. This is primary structure. Now coming to secondary structure. So secondary structure happens when there is a further folding of primary structures, okay. So now see, secondary structures are of again 2 types. One is alpha helix and in the beta pleated sheets, okay. So now what happens, this you have seen that chain, right? There is a polypeptide chain. This polypeptide chain, if it is having it is like coiling, okay, helical structure. It can, this polypeptide chain will coil around, okay. That will be called as alpha helix and that is called the secondary structure, okay. So now what happens here? Due to the folding of the backbone of the polypeptide chain due to the hydrogen bonding between CO and NH group of the peptide bond. So I will tell you what is this. I will explain this with the diagram, okay. Now what happens in beta pleated sheets? In beta pleated sheets, the polypeptide chains laid side by side are held together by intermolecular hydrogen bonds, okay. We will see the diagram and we will understand. See here, okay. Now this is one polypeptide chain, this is one polypeptide chain. Here what has happened, the polypeptide chain has coiled around, right, and there is a hydrogen, like in one turn this NH group is there and here this CO group is there. So there is a hydrogen bond here, right. Similarly, in beta pleated sheets what happened, this sheet, this one is there and this is another sheet is there, okay, and there is a hydrogen bond between the two sheets or two polypeptide chains, right. So that is called as beta pleated sheet structure, okay, clear. In alpha helix what will happen? Coiling will take place. In beta pleated, the two polypeptide chains will lie and there will be hydrogen bondings between them, okay. Next, coming to the third one, that is tertiary structure, okay. Now what does tertiary structure is there? Tertiary structure represents the overall folding of the polypeptide chains, that is further folding of the secondary structure. So we have seen first there is a plain chain of amino acids which we call primary structure. Now this amino acid if starts coiling, then it becomes one type of secondary structure, that is alpha helix. Now if there is a these two like these polypeptide chains are put in sheets and they form some high bonding between them, then we call it beta pleated sheets, okay. Next, if we see, the tertiary structure exists in two different molecular shapes which we have already discussed, fibrous and globular, okay. So that alpha helix will coil around and form globular proteins, and this beta pleated sheets form fibrous proteins, okay. Next, you can see these tertiary structures are stabilized by hydrogen bonds, disulfide linkages, van der Waals forces and electrostatic force of attraction. So please remember, this point is important. We have hydrogen bond, disulfide, van der Waals and electrostatic. Hydrogen bond, van der Waals, disulfide and electrostatic attraction. Next, coming to the quaternary structure. So in quaternary structure, more than one polypeptide chain is involved, okay. The example of quaternary structure, you can see, example of quaternary structure is hemoglobin, okay. So next, coming to, we can just see all these structures once in detail. So if you see in primary structure what we have done, these are the different amino acids, right. Single amino acid, one straight chain of amino acid is taken, and that is called as your primary structure. Now these amino acids can coil around, okay. These amino acids will coil, this polypeptide chain will coil, that will give you alpha helix, or it can be present in the form of sheets, we call it beta pleated sheets. Now what happens in case of tertiary structure? There is further coiling of these amino acids, polypeptide chains further coiling, and we get tertiary, and here we have two types, that is fibrous, okay, and globular, okay. Next, coming to quaternary structure. So in quaternary structure what happens? We have further coiling of like more than, we have here more than two polypeptide chains, right. So they coil around to give you another structure or more advanced structure, that is your quaternary structure, okay. Next, coming to denaturation of protein. So to understand denaturation of protein, first we need to understand what is native protein, okay. So when the protein is having a unique 3D structure and biological activity, okay. So every protein has a unique function, a unique structure, then that protein is called as a native protein, clear. Now what happens? Whenever there is some physical change like the change in temperature or the chemical change like the change in pH, the hydrogen bonds are disturbed. So we have seen there is like the protein structure is stabilized by hydrogen bonds, right. So whenever there is a physical change or there is a chemical change, the hydrogen bonds get disturbed. So this if there is a globular protein, then it will start unfolding, and if it is a helix, it will start uncoil, and then protein loses its biological activity, okay. But here you should remember that the primary structure is not affected by the denaturation. Example of denaturation: coagulation of egg and curding of milk, okay. So here is an example. This is egg, and we have here, this is the structure of the protein. Let me come here. This is the structure of the protein. When there is denaturation, that is there, when you boil an egg, the proteins get denatured and it becomes the primary structure comes, and that is not disrupted further, okay. Fine. So That is all about proteins. Now we'll discuss something about enzymes, not a very big topic. So enzymes are biocatalyst, means they catalyzes the biological functions. Then you have generally they are globular proteins, okay, not mostly they are globular proteins, but not mandatory, everyone will be globular. Then they are very specific in nature, so these enzymes will react with a particular substrate and for a particular reaction only, okay. They reduces the activation energy. Like for example, here you can see, suppose this is a suppose this is a you have a substrate or enzyme, so this site is there, right. So this is an enzyme, suppose this particular substrate only can react here, this particular substrate can react here. If you see here, this substrate only can react at this particular site and this one can react at this particular site, okay. So only certain substrates can react with certain enzymes, that means this enzyme cannot react with this substrate, okay. That is called about the specificity of the enzymes, okay. So yes, now we'll do some questions here. Okay, last class one question I gave you, the glycosidic linkage involved in linking of glucose units in amylose, okay. If you remember, amylose was C1C4 alpha linkage, option number D, okay. Next, proteins are found to have two different types of secondary structures, that is alpha and beta. Alpha helix structure of the proteins is stabilized by. So if you see, we have just discussed now, right. I told you to remember alpha helix are stabilized by hydrogen bond, okay. So here the answer will be, you should remember, hydrogen bond, okay. Next, alpha helix structure of the proteins, okay, this question is repeated, okay, fine. Next, which of the following is optically inactive? So if you remember glycine structure, okay, so glycine has like this, C double bond OH, NH2, and it has two hydrogens, okay. Now this particular structure is called as glycine, and because of two hydrogens attached to the same carbon, this becomes optically inactive, and correct answer is option number A. Next, which of the following amino acids is aromatic in nature? So now let us go back and see the structure of these amino acids, if you can recall the structures. So certain amino acid structures only you need to remember, okay. So we have like, if you see here, yeah, histidine is there, which is aromatic, right. You can see the structure of histidine, okay. This group is there, it can undergo resonance, and that is why this is a aromatic amino acid, okay. So we have tyrosine also. Okay. Now let us see the next question, question number 4. Which of the following amino acids are aromatic in nature? So histidine is aromatic in nature, okay. Next, non-essential amino acids. So if you have remembered essential amino acids, then easily you can find non-essential amino acid, and that is your glutamine, okay. Next, neutral amino acid. So we have discussed acidic, basic and neutral. Glycine, if I see the structure, glycine has no other NH2 or COH group, so that is why it is a neutral amino acid. Next, correct statement, peptide bond is formed by the loss of water molecule. Yes, that is correct. So option number A. How many peptide linkages does a hexapeptide have? Hexapeptide means there are 6 amino acids, right? 1, 2, 3, 4, 5, 6. So how many linkages will be there? 5 linkages, option number B. Proteins are polypeptides, which is greater than 100, right? Next, which of the following bonds is not found in fibrous protein? Peptide bond is there, hydrogen bond is there, disulfide is there, phosphodiester is not found in fibrous proteins. Next, which type of bonds govern the secondary structure of proteins? So secondary structure is governed by hydrogen bonds. Next, fibrous and globular are classified on the basis of secondary structure, right? On the secondary structure, we have fibrous and globular proteins. Next, which of these statements is true about the secondary structure of proteins? Secondary structure of proteins occurs only in the form of helices. No, it has alpha and beta pleated sheets. Protein helices are always double stranded. No, that is also not there. Protein helices are right handed. Yes, this is correct. Okay, all the protein helices are right handed coiled. Okay. Next, Hemoglobin belongs to which level? Tertiary, sorry, not tertiary, quaternary. Okay, option number A. Okay, next. Okay, next question, in which of these structures of proteins can disulfide bonds be found? So disulfide bonds can be found in your tertiary structure. Okay, option number A. So here's a homework question for you. What makes up the primary structure of protein? Okay, very simple and basic but fundamental question. Okay, please answer this question in the comment section. So students, we are done with the part 2 of biomolecules. I hope you have liked this video, and if you have found this video helpful in your KCET preparation, do not forget to subscribe the channel. Okay, thank you for watching, and see you soon in the next episode where we will be completing the biomolecules chapter. Okay, all the very best for your KCET preparation.